Welcome to this first session where we're going to discuss some key outcomes from the Solace project by talking to different partners who are involved in organizing and running a series of online stakeholder workshops where many of the key messages and results from the Solace project were addressed. So to start off, we had an entire series just about roots because a lot of people here are very happy to talk about roots. To start off with, the first event in the series was about deep rooting, and it was hosted by Christian Thorpe Christensen from the University of Copenhagen, so welcome, Christian. The second event was about root phenotyping, hosted by Xavier Drey from the Université Catholique de Louvain, so welcome, Xavier. And the final event in the Roots series was about root and plant microbiome interactions, hosted by Christophe Salon from INRE, welcome. But like the Solace project, the events didn't just focus on roots. There was also a fourth event about how microbial inoculants and biostimulants are evaluated and deployed in controlled and uncontrolled environments. A bit of a long. We called it the Upscaling Knowledge event, hosted by Julia Cooper from the University of Newcastle. Welcome, Julia. And last but not least, Tim George from the James Hutton Institute hosted an event about using modeling to aid in decision-making for agricultural policy in Europe. So welcome, Tim, as well. And before I start, I also want to say a big thank you to Amelia, who unfortunately couldn't be here today in person. She coordinated all of these online events, as well as this session and much of the content today, so thank you, Amelia. So let's get started. First of all, we had a whole series on roots. Clearly a lot of energy in this project went into roots. No pun intended. Xavier, maybe you can start off by telling us why roots.
Thanks, Laura. I'm taking my notes to be organized and stick to the time. You know. Well, plants grow with resources that they acquire in the environment. And the main resource they depend on are light, CO2, which they get from the atmosphere using leaves, and on the other side, water and minerals that they acquire with their roots. Now, the acquisition of each resource is a completely different story. For example, if you take light always comes from above, and it's more intense at noon. So plants have learned to unfold leaves high in the canopy when they need more light. And breeders have learned to relied on these simple rules to optimize the capture of light. Now coming to soil resources, they are not always available in the same layers of the soil, in the same amount, and at the same time. So in other words, plants need to find ways to adjust continuously to soil resource availability. So breeders are struggling to make varieties that are good at doing that. So we seem to be left to apply large amounts of water and nutrients to the field. And if we believe that a resilient agriculture relies on reducing such inputs, then all actors, farmers, scientists, industry, policymakers, funding agencies, needs to pay as much attention to the root that they have been paying to the shoot part. Thank you.
That's a great way to start us off on roots. Thank you, Xavier. Christian, the somewhat cheeky title of your event was Too Deep or Not Too Deep. So is it valuable for crops to have deeper roots, and why?
Yes. Okay. Yes, we think it's very important, and I think that the first reason is super simple. I mean, if you have a crop with roots to 1 meter's depth, then it can assimilate resources down to 1 meter. If you can have a crop with 1.5 meters roots in depth, then there's a 0.5 meter extra resources to tap into for the crop. So in that sense, of course, in real life it's not that simple, but that's a simple principle behind it. And especially water and nitrogen, they tend to move in the soil and move deep. So especially for these resources, deep rooting is really important. And I think it has been known for many years that in dry conditions you want deep roots for the plants to be able to rely on deep water when the soil is dry up.
So in that case, how do we achieve deeper roots, and what are the traits that can be selected for?
I think, there are many ways to achieve deeper rooting, and of course plant breeding is the focus here and is an important one, but also management. Things like sowing time has a big effect on the rooting depth of a crop. Things like crop rotation, and in some cases by choosing a crop that has deeper roots and an alternative crop, so there are different possibilities for that. And the trait, that's part of what we have been looking for in this and other projects. What traits can we look for? And of course, we can look for the deep roots themselves, but also that is a huge work and very difficult. So we are also trying to look for other traits that relate to deep rooting. And that can be things like, just an example, drone flying thermal imaging, looking at canopy temperature that shows whether a crop is drying out or not, for instance. But there are many other attempts of simpler measurements.
Thank you. Moving on to Xavier, the event you facilitated was about root phenotyping. So for those of us who aren't quite the experts that you are, what is root phenotyping, and how is it different from looking at roots in the field?
Well, it's an interesting question. Well, I like to describe root phenotyping as an art. The art of observing, describing, and quantifying plants. So for root phenotyping, we observe cells, tissues, roots, root systems, plants, and sometimes field plots. We describe and quantify the shape, the architecture, the properties, are they conductive or not, the processes, do they acquire nutrient efficiently or not, the performance, and the interactions between plants and their environment, and also their adaptability. As I mentioned, they need to adjust consistently. So ideally, we would prefer to observe roots in their natural environment, as we do for stems, as we do for leaves, as we do for flowers and fruits. But this is far from easy, because we just can't remove the soil around the roots to see them. So scientists are forced somehow to use artificial cultivation system in pots, in hydroponics, and so on, to make the observation possible. However, on artificial system, we get kind of artificial plants. So we need to be wise and cautious when we conduct root phenotyping. Fortunately, the modern techniques that we have in artificial conditions gives us so nice descriptions that we learn a lot, even on artificial system. And so, even if acquired in artificial conditions, this new knowledge can be used to develop new varieties or innovative management, which was the topic of the project.
And what are you looking for specifically when you use these types of platforms? Are all roots equal?
Well, phenotyping comes, as I say, it's not a scientific discipline. Phenotyping comes in support of plant scientists and industry. So for scientists, the aim will usually be to uncover features or behaviors that will make plants more able to grow and to produce under low input conditions. Probably the objective is different for industry, where the aim may be to quickly identify plants that possess these features, or to test products that support the plant in doing that. But these are rather conventional examples. Phenotyping is evolving quickly with technology. It's more and more connected to big data world, and so novel and smarter ways to use it appear every day in the literature. So you ask me if all root systems are equal. Well, what we learned from root phenotyping is that root systems are not similar, and they are not equal. They are extremely variable in their constitution and also in their performance. This diversity, well, it's an opportunity, because we learn a lot when we see big differences. But what we also learned is that the genetics of root feature will be complex, that there are different ways to get to the same level of performance, as Christian was saying, and that what makes a plant efficient to acquire water in some circumstances can make them at the same time inefficient to acquire another resources. So we are playing with constraints. So they are not all equal.
Thank you. And Christoph, your event was cleverly named To Deal or Not to Deal. It focused on plant-root microbiome interactions. What is the benefit of having roots that communicate with microbiome?
Oh, I don't know how it works. Okay. Well, we know all that plants interact with plants so to cope with nutrient limitations, also to fight pathogens and so on. So we know also that modern breeding programs have neglected plant microbe interactions. But however, there are genetic and physiological determinisms that will allow plant and microbiome interactions to help the plant to go further in the performance. And as an example, I could quote the nutrient efficiency. First, we know that most of the plants, most of the crops establish symbiosis with mycorrhiza, for example, for water or phosphorus acquisition. We also know that for grain legumes, because I'm a legume maniac, I'm sorry, that grain legumes establish symbiosis with rhizobia to get the dinitrogen from the atmosphere. And as such, we can decrease the burden of fertilizer and we can participate to the mitigation of climate change. But besides symbiosis, there are other microbial communities which benefit the plant. And during the speeches we had with the stakeholder events, I showed some work that has demonstrated that besides symbiosis, other microbial communities are key for fighting stresses, allergic stresses, for example, drought stress. When you use a mutant and which establishes no symbiosis, you see that his performance is better when the diversity of microbial community besides symbiosis are improved. And lastly, besides carbon, nitrogen, you have other nutrients that the microbes could help the plant to cope with. I'm thinking about, for example, iron, and you know that microbiota can improve iron nutrition and is a lever to fight iron deficiency. And as an example, you know that purverdine, siderophores, which are help to cope with iron deficiency. And it has been demonstrated in Arabidopsis, and in somewhere else. So many benefits.
And what kind of root traits and other plant traits encourage this type of interaction?
Well, again, if you go from mycorrhiza, nodules, and all the other traits, for mycorrhiza you could perfectly try to look at the traits that host the interaction, so the hyphae. Going back, you could simply work on the root architecture, work on structural traits that will describe your root architecture. It can be simple traits like the length of the different roots according to their typology. It can be the diameter of the roots, the location of the different roots on the root system. Coming back to the structure which hosts the interaction, when you work on nodules, for example, for grain legumes, you can characterize the nodule of the grain legumes, the size, the shape, even the color. For mycorrhiza, this is the same. And also, I was talking only about structural traits, but there are also functional traits, which is trickier. For functional traits, you could work at the link between plants and microbes. And I'm thinking of exudates, which are the signaling and trophic links between plant and microbe. So many traits, some easier, the morphometrical, and some trickier, the functional.
And how would you evaluate the benefits of root microbiome interactions?
Well, you could go from simple evaluation on plants alone, for example, just looking in your control condition or in your field at the biomass of the plant. It's very straightforward. The elemental content. You can go further looking at the genome content of the plant. You can also assess, again, talking about legumes, sorry, and nodule, you could work on the health, the pink color of the nodule, which is an indicator of the health of the nodule, but also on the size and number of nodules, because we know, I can't do the equation, but we know that the flux of nitrogen which is entering into grain legume is proportional to both the number and biomass of the nodules, but also the specific activity of the nodules, which is the ratio of the flux by the biomass. Well, you're lost. If you want to go further, at the longer term, in European project, we are talking about impacts of and so on, you could assess the benefit of plant and microbiome interaction in the decrease of fertilizer use, the decrease of associated detrimental effect on the environment, pollution, greenhouse gas emission, the benefits on soil fertility, on biodiversity. So this is the longer term and
of course, these types of evaluations are different in field versus controlled conditions, which comes nicely to the next event, the workshop that Julia ran on Upscaling Knowledge, which used biostimulants and microbial inoculants as an example. So, Julia, can you tell us about some of the discussion you had about the how to address the difficulties of evaluating microbial products in controlled conditions versus on-farm?
Yeah, I mean, we had a workshop where, in the Solus project, we actually evaluated microbial inoculants. I guess you could consider 3 situations. So we had sort of very controlled conditions, like in sort of glasshouse type of situations. And then we had field experiments that were still controlled field experiments. And then we had sort of more on-farm experiments as well with the microbial inoculants. So we tried those three different ways of trialing them. And we experienced the usual challenges of not always seeing benefits or positive effects as we moved from the more controlled situations to the less controlled situations. Yeah.
Before we get ahead of ourselves, maybe we can take a step back and you can tell us what exactly or how you define biostimulants and what was evaluated in the Solus project that falls under this category.
Yeah, so we use this word biostimulants, kind of a very broad term, and I Googled it. I mean, I do know, believe me, I did know without Googling it, but I just wanted to double check the specific definition in the 2019 EU Fertilizing Products Regulation, where it's substances and microorganisms which can stimulate natural processes to improve nutrient uptake, nutrient efficiency, and crop quality. So in Solus, we really were looking at the microorganism bit of that, not so much the substances, which might not be living organisms. So we were looking at microorganisms, plant growth promoting rhizobacteria, and fungi, particularly AM fungi and trichoderma. So these kind of products that have been isolated in laboratory conditions and then multiplied for application in the field. Yeah.
Okay, great. So you've mentioned some of the challenges and also that it's not always coming out the same in the controlled and field conditions. But what about Solus's focus on trialing microbial inoculants? Do they work? What conditions did they work in?
Do they work? Yeah, as always, I'm going to give you a scientist's answer and say it depends, right? We got mixed results, and I think Philippe showed some of those on his first slide. So there were some cases where it did seem to work. Arvalis in France trialed, I think it was AM fungi, and found under some conditions positive effects. Particularly when there was more nutrient stress. Other cases we didn't see positive effects, and then we had Gunther Newman contributing in that workshop, and he provided some information sort of from wider studies he's done, and he really elucidated that link between soil conditions, particularly soil carbon. There may be a link. So soils that are actually less fertile, less healthy, may respond more when you add an inoculant. But then there was also this other issue of if your soil is too poor and too infertile, then that inoculant may not get established. So basically there's still a lot of questions to be answered around soil conditions and the microbial consortia and management practices really, and how to put those all together to make them work in the field. Well,
that maybe leads nicely to the next, the last of the sessions that we had about modeling. So when we think about modeling as a decision-making tool, what do policy stakeholders see as the key benefits and limitations, Tim?
Okay. Can you hear me? Yeah. So, I mean, basically all these phenotyping techniques and experimental approaches are all great, and they've yielded some really great results. But what the modelling allows us to do is to take that beyond the confines of the experiment, beyond the confines of this particular situation. I think that is what the decision makers really find useful from the modelling, is that ability to be predictive, to predict the future, to predict the impact in different environments. And they also really like the fact that the modelling often comes with very visual results, so something that they can really see very simply and don't have to know the detail about. So for the decision makers and the policy makers that were involved in our stakeholder engagement event, they say that modelling is really useful for them. But it's one source of information. They really stressed that it's really one source of information. It's not the be-all and end-all. They were particularly interested in the solace modelling outputs, which highlighted this trade-off between farmer income and environmental stewardship. So really highlighting the fact that you can perhaps not have both, and we need to work out the best ways to achieve optimal conditions for income versus environment. But they also really made the point that that the modelling should really come with a health warning, and we need to be upfront about what the limitations are of the modelling and what the caveats are, because if we don't tell people that, then there's a real opportunity, there's a real potential for the modelling results to be taken out of context or to be taken beyond their capabilities. So I think that was really a really important point.
You've touched on this a little bit already in your response, but maybe just to clarify, can you tell us what kind of modelling we're talking about in the Solace project, and what you discussed in terms of that in the event?
Sure.
Modelling.
So modelling, there's the life cycle analysis or LCA approach, and then there's a crop modelling approach, which is a mathematical modelling approach. There are also some root modelling approaches in package 2 as well. But both of these need inputs from the data that we produce. So the data validates and sets up the models nicely. The LCA, or the life cycle analysis, this really allows assessment of costs and benefits of interventions in the current state. So we can look at the impact of, say, one of the interventions put forward by science, let's say the biological inoculars. We can look at the impact of that on the economics of the system, on the environmental impact, but also the yield and so on. We can predict how that has impacted in different situations. Whereas the other modelling, the crop modelling, or the DSAP modelling as we call it, this allows prediction. It allows prediction into the future. It allows us to predict the impacts of climate change on the growth and the production of crops under different climates and with different interventions, let's say fertiliser, reducing fertiliser amounts or so on. So two really clear, different approaches, but both useful in their own way and both interactive.
And you also mentioned before farmer income and environmental stewardship. What are the trade-offs between the two, and how do we evaluate the impact of agronomic interventions when it comes to policy, especially if some of the scenarios modeled in Solus indicate that we may need to rely on one particular intervention more in the future?
Sure. So, I mean, as I said, one of the key findings from our workshop was the interest in the fact that we were able to demonstrate the trade-offs between farmer income and environmental impact. So we're able to demonstrate from the life cycle analysis, for example, that although we can maximise impact by adding nitrogen fertiliser, maximise income by adding nitrogen fertiliser, we're increasing the environmental impact by doing that as well. So a trade-off between those two is really important. Some of the things that really came out though from the workshop were that we need to make sure that we assess other interventions that are of interest to the stakeholders. So things like the impact of rotations, the use of legumes in systems, as Christoph talked about, cover crops, tillage, organic agriculture, precision agriculture, but also things like irrigation. A lot of the outputs of the Solace modelling show that we'll need to irrigate crops in the future. So what are the environmental impacts of that? Also, it was highlighted that there's a belief that there's a real opportunity to look at some specific things as well, like the trade-offs involved in the use of potatoes in regenerative agriculture, for example. So we have some opportunities which we can pick up later in the classroom session later. But one of the things that a lot of the stakeholders identified was that we always need to be mindful of the impact on the major global issues like biodiversity and greenhouse gas emissions, and we need to have those as outputs to our model as well.
Thanks. So we've touched on a range of scale here, from roots and root microbiome interactions to in-field assessment and evaluation and modeling that I think covers over 30 years. So, Christian, how do we combine these different disciplines and scales, and how do we approach crop improvement in preparing to address climate change? Just a little question for you.
Yeah, and no, that's a very big question, and I think Tim just said some of it. I think the modeling is needed. We have a complexity that is so huge. We have different crops, different rotations, soils, management practices, and all of it subjected to different weather and climate and so on. So the complexity is huge, and if we want to take, you can say, the results we have from some conditions and make good quantitative estimates of what will happen in another situation or upscaling to a national level or something else, then we need the modeling. But we also need to remember that models are not better than the science we put into them. And I think it's important also that we keep in mind we need everything from the detailed studies for some of the processes going into the models, but we also need what Julia just said. It was difficult to take it into some of the field conditions to at least study some examples of this complexity to make sure that our models are doing reasonably well.
And how do you include roots in this, especially since they're not as visible to farmers and breeders?
Yeah, but that's what we try both by measuring the roots themselves. We are trying to do dynamic measurements on root systems, so we can actually put into the models how fast will they grow and how will that depend on a number of things. But also, I think from experiments both in this project and others, data that show, when you have a crop growing and it has a root growing, how does water disappear from the soil as the crop uses it over time and from different layers? The same thing with nitrogen. So we actually get some. Even though getting root data and soil data is always difficult, we need to have a certain amount of them to make sure that our models is predicting something meaningful. When I started to look into roots and modeling, I guarantee you the relationship between what the program code was predicting and what the roots were doing was not good.
Great, thanks. Xavier, something else I've heard a lot about in the project is high throughput phenotyping. So what does this mean and how do you do it?
So high throughput basically means the ability to handle hundreds or thousands of plants. That's what it means. So the initial motivation or the initial need for high throughput comes from genetics. Remember what I said before. We learned from phenotyping that the genetics of fruit features is complex. So it's not like the green and yellow color that Mendel was playing with a long time ago. And complex genetics comes with the need to observe and manipulate thousands of plants before we are able to identify which parts or which genetic factors are carrying the information that will help the plants to behave the way we would like it to behave. So you may have heard also about plant phenotyping platform. So it becomes a common name. These are large-scale phenotyping installations that can handle these numbers of plants. And we had some that were used in Solis. Europe is also encouraging different platforms and countries to work together to provide a more comprehensive service of phenotyping. So this is the Emphasis initiative that maybe some of you have been already working with. Now with the time going, high throughput has also taken another meaning, that of being able to follow the plant over time and to see how it reacts to subtle changes in its environment. And that's a more recent development in high throughput phenotyping.
Thank you. Christoph, do you have something to add to that? Why use high throughput phenotyping?
Well, thank you, Xavier, because you gave a lot of an already quite complete answer. Well, I could add that plant phenotyping platform, either in the field or in control conditions, are highly instrumentalized. And to realize what Xavier was saying, you need sensors, you need imaging facilities, you need to record not only the phenotype of the plant, either structural or functional again, but also the environment, the climate and microclimate. So, high throughput plant phenotyping platform, whatever in the field or in control condition allows to do this. I could make some comments about to compare field platform to control condition platform, which are not the same power for specific objective, but it's roughly the same. So you can work with thousands of genotypes, with many replicates, with mixture of plants, with association of plants, with whatsoever you want. And it's dynamically, as said Xavier, with high resolution according to the platform. You can get images up to 7 microns in some platform. And also the use of phenotyping platform allows you with imaging and with the fact that it's non-destructive to increase the statistical power of your research. And lastly, with this, what do? You can use in control conditions very close to the mechanism to assess the determinism of the mechanism, to link it with a trait of interest, to work on the interaction between traits, and as such to understand better the functioning of the plants. And lastly, well, you could say common sense, phenotyping platform. This is all. We are always saying this when you want to get some money from the grants. Helps you to speed up the selection of plants which are more tolerant to abiotic or biotic stresses and to save the world.
Save the world, good. I know that modeling also plays a role here. So Xavier, what's the relationship between high throughput?
Grow like plants, they have leaves like plants, they have roots like plants, but they exist only in your computer memory. So one advantage of these kind of plants is we can grow virtually hundreds of them in hundreds of different environments in 2 minutes time. And so the process of scientific analysis can be much faster if we include modeling. And in particular, the way we use it mostly is that virtual plants will help us to test the limit of our knowledge. If we bring in the same virtual world what we know about soil hydrology, what we know about water acquisition, what we know about photosynthesis, and what we know about growth, then we can come out with a specific prediction, and these predictions, a long number, we can test them in phenotyping experiments. And if it turns out that these predictions are not realized in reality, then we know that there is something missing in our knowledge. So modeling and phenotyping are part of the same loop.
But this is all very virtual and in that sense not entirely real. So how do you improve trust in these in this modeled data?
I was expecting this question. Obviously, virtual plants are virtual. So they are only a simplified representation of reality. And again, we need to be wise, as we are wise with artificial conditions, and it's important to continuously reconnect modeling and reality. So the aim of modeling is not to copy a plant. The aim of modeling is to capture the essence of what is important in a certain context to achieve a certain level of performance. So the model must be kept simple. If it's not, if we try to put too much into the model, then it's likely that we end up with a tool that has so many buttons that it's always possible to turn the button and find the solution that you want. So it's not helpful at all in that case. So I would say from my perspective, the work package 2, is that a virtual plant model will be trustable if it turns out to be generally useful. And this is probably different from the perspective of a model that is used for taking decisions.
Well, that leads very nicely to the next question. Tim, your event specifically asked policy stakeholders to use modeling in decision making. So same question to you.
So, I mean, essentially, there were three main messages that came back from the stakeholders in our session. And one is that it echoes what Xavier was saying, which is we need to promote user confidence. So the users need to be confident in the outputs. So this can be achieved in a number of ways. Co-creation with the decision makers. So the decision makers are there from the start, they know what's involved. They suggested that examples of real-world scenarios, so demonstrating that the model works with known scenarios. For example, the impact of the recent spike in oil prices might have a big impact on fertilizer prices, etc., and what impact that has on the economics and the environmental impact, etc. So giving them those real-world examples gives them confidence in using the models. They also suggested that they would like to have a user-friendly interface, so they talked about a plug-and-play approach, which is essentially where they have the opportunity to use the models themselves to see what the impacts are. Pressing all the buttons, as Xavier said, but maybe limiting those to the ones that are relevant. The second message was that modelling should provide interventions, a range of interventions for policymakers to use rather than being particularly prescriptive. So giving people options to give advice rather than prescriptive. And ultimately, this should all lead to the models helping to create proactive policy, they were saying, rather than reactive policy. So what they want to be able to do is to proactively put together policy which allows them to cope with major issues in advance rather than reacting to them as they come forward. So.
Great. In the interest of time, I'm just going to skip ahead a little bit and ask one more question to Julia. I think that this question of trust seems relevant across a lot of different experimental research. So in terms of microbial inoculants and biostimulants, relaying the benefits of these types of products to farmers can be tricky, especially, as you said, since the results don't always come with very clear answers. So how do you build trust in experimental results, and similarly or related, how do you encourage farmers to participate in on-farm experimentation?
Yeah, I don't know if it's about building trust in experimental results, or, I think ideally we want to help farmers to critically evaluate new products and innovations that are introduced to them. And so in our project, in this project, we realized that that's quite a important thing, that just simple things like encouraging farmers to trial innovations side by side with a control, with their standard practice. And I was thinking, the reasons that farmers get involved in experiments and trialing things isn't necessarily the same as the reason that scientists do, or that scientists even want to work on farms, right? So farmers are more interested in, on my specific farm, is there a positive effect that actually has an economic benefit to me? Whereas I think within Solace, a lot of us were more interested in seeing, in general, across many, many farms, is there a statistically significant yield effect? It's not quite the same thing. So one lesson I think we might want to take away from Solace is being really clear on what the scientists want to get out of the involvement and what farmers do. And just keep that in mind and just sort of together, which the next session is going to be about how do we better kind of co-create research activities and questions that meet the needs of both both parties involved.
Great. Excellent segue, Julia. Thank you. So thank you all for your input and thanks for your attention. Of course, we've just scratched the surface of what was covered in these events. They'll all be available online at some point in the near future as well. And hopefully they'll provide some input for the session this afternoon as well. If you have any questions, of course, if you're here, feel free to approach our guests during the break. And again, as we mentioned for the online participants, there's also the Klaxoon board where you can add your questions. So thank you again to everyone for participating. And